Hi, everyone. Thank you for bearing with me for those few awkward moments. Hopefully that's the most awkward it gets in this talk. I am Beatriz Costa-Gomes, and I'm a research fellow at the Alan Turing Institute. And we work with, and it's the Institute for Data Science and Artificial Intelligence. So yeah, I'll be talking about machine learning. Hopefully it's exciting and you find it exciting. This is a work in progress. I want to show you what we've been working on in collaboration with some people that are here. I'll mention them in a second. And we really want to continue to develop this tool, and we really want to get some input from you guys, and we're really excited about this. So why are we developing a tool? This is the main question. And the truth is that we've seen all of these talks, and they've been really great. And a very important thing is actually to find and reliably detect the structures, right? Like you need to somehow get the structures out of this image that you get. And I have this image here that Marston Bradshaw from the University of Bristol, he's the one that provided the data for me. And you can see all of these filaments. So the question is, can we get these filaments? And what can we learn from the data that you guys have already have? This is what we are trying to do. We don't want to create more work in the sense that you have to do more data or analyze it again. Can we use the data that you already have and actually get new structures from it, or other structures, or even just oddly shaped objects that you had to previously hand annotate, and we tried to get it in a more automated way. So this is what we're trying to do. And the idea is that we go from an input image where you've had your detections, and we can actually individually detect the different objects in it. In this case, it's filaments. And in each color on the right side, you can see that would represent a different object. So for this, we developed GRACE, which is the graph representation analysis for connected embeddings. And this is more or less the workflow that we have, and then I'll walk you through a little bit. But before I get to all of that, I guess the first question is, what is a graph, right? And why would we want to make graphs out of these images? So graphs are basically organized structures. You have these things that are called nodes, and they are connected. So that's what it is. And it basically describes the higher organization, and you have like this local context to each of these individual nodes because they have neighbors, they have connections, and you can have paths in them. And the thing is that graphs are everywhere in nature. A molecule can be a graph. In this case, this is caffeine, my favorite molecule. You can describe the organization into filaments as graphs because you have different filaments that are connected. A brain, so the connection of neurons are actually, again, another graph. Social networks are a very good example. We're just connected to each other. It doesn't have to be a physical connection. And even the tube, basically, is a graph where you have the stations and you have the connections between them. So graphs just allow you to get all of this information, all of this structural information. And this is what we care about. So the input is these images. And what I've asked Marston to provide us is actually this side, a very dirty data set of detection. So this is just normal detections obtained with Criollo with a threshold of 0. So there's 0, some of these detections have 0 confidence. And this was trained on actually trying to find these proteins attached to the filaments. So it was trained on trying to find the proteins on the filaments, and then we get all of this with 0 confidence if needed. This is the whole thing. But we have this very big image, right? This has for almost 4,000 by 4,000 pixels. And can we reduce how big this data set is into actually more usable things? So the first thing that we do is actually look at each detection as an individual image. So just in that, we reduce the images from having all of these millions of pixels to just having a few hundred or a few thousand. And what we want to do in this step is actually to get a description of these images. And we can look at that and say it's a green blob. But even if we went way more than that, the computer wouldn't understand. So what we want is actually to get this extraction. So the idea, as I said, each detection box is treated as an individual image. And then we put it through this neural network. And the idea is that we have a pre-trained neural network. This is off the shelf. We just use the ones available on the Python libraries. And we don't care about the classification. We just care about this last step there, where what happens in a neural network, especially like something like a ResNet or a convolutional neural network, is that your image gets described through all of these layers, and the information goes from one layer to the other, but it goes in a format that the next layer is going to understand, and it's going to learn with that information. So what we get at the end is actually a vector that tells you all of the features that this ResNet or this network or any network would actually use to classify the image into what it would be. We don't care about the classification, but a good analogy is this would be all of the lines that you would use, for example, to this. If you have an image of a cat or a dog, you would describe, for example, how the tail looks or the hair looks. So you would have a vector of all of these descriptions. This is the idea what we're trying to get. And why we want this is that we want to take these nodes and these description of these images and actually make a graph into it, where the number of nodes is actually the detections. So an easier way to see this is that instead of considering the whole image, we actually consider all of the detections and we connect them. In this case, it's just the simplest triangulation that we could do. It's just a triangulation with no crossovers. And each of these nodes individually cares about the detection, has the spatial location of the detection, but also has all of these nodes that we learned on the previous step. So now we reduced this image space into a more structured and organized space. And now what we want to know is, can we retrieve objects from or smaller structures from this organized space? And this is when we need some input from you guys, because we need to know what we are looking for. So this is the only part where we need some annotations from you. And you're like, more annotations. Sorry. We do need to know what we look at. And this is a widget that we built for Napari, which is an ND viewer. And the idea is that you just, okay, now it's playing, right? Yeah, you can just see all of the detections, all of the nodes, and you just make an annotation on what you consider to be an object that you care about. In this case, we want to find the filaments. So we just annotate. And when you do this, it basically, as you can see, they change the colors, and what's doing is attributing a ground truth. So the nodes inside the object are considered 1, the nodes outside the objects are considered 0, and the edges that connect things inside an object are considered 1, and the edges outside of the object are considered 0. So we get this ground truth. This is what it looks like. This is just a random annotation of what an image would look like. This is just to exemplify how the annotation would be at the end. And I want to just show you the result of this. For example, if we care about the node annotations, that all of the ones on the lines would be considered 1, and all of the ones outside of the lines would be considered 0. So this, we get a ground truth. And what do we do with this ground truth? So what we actually do is that we're going to look at this as subgraphs. So basically a smaller graph inside that big graph. And we're going to classify this subgraph. And what way do we classify this? So basically we take each node, and we remember that the node has the true annotation at this point, and it has all of the features retrieved from the extractor. So it has the description, and it has what that description means, and the computer understands that. But we also take the neighborhood. And what we do is that in this case, we just care about the direct neighborhood, so only one jump. And what we do is that we bring another network, we bring a graph convolutional network, but in this case, we actually care about the classification. So what we do is that we feed these subgraphs into it with the annotations, and we try to get, basically, we want to learn the relationship between the nodes. So basically their shapes, how do they connect to the different nodes around it, and also all of the annotations and all of the values around them. So by the way, these subgraphs are all belonging to this graph, even if they have odd shapes. And then what we get is actually a probability of each node belonging to an object or not. So even if we have sparse annotation, we train this model to say, look, there's a probability that this node belongs to an object, this node doesn't belong to an object. But this probability is still not great. We still have a lot of, there's still a lot of space for errors. So for this, we actually care about a compiler, an optimizer, because we are going to use it to compile all of this information of the probabilities of the edges and the nodes, and we actually are going to compile them into different objects and separate objects. And this is the part of the pipeline that we're still working with synthetic data. Everything else, as you've seen, has been with real data. Now it's the only part with the synthetic data. And the idea is that we have these probabilities, the probability of a node being an object or a link existing inside an object, and we build this hypothesis. We have some combinatorial search, and basically we get a solution that gives us the most probable solution of each object. Okay, and basically what it does is that it goes through the graph, through all of these probabilities, and tells you what are, and chooses the one with the highest probability of these objects being what they are. So if we look at filaments, for example, in this sparse data, or this dense data, in the dense one, you can see that the filaments are quite well defined. But in the sparse one, even though the nodes are quite far apart, it still learns some of the objects, and some of them are still separate, and I'll get to that. But the idea is this. But the most exciting thing is that with this optimizer, what we can actually do is find all of these odd shapes at the same time inside your data set, inside your image. So we care about filaments, but we care about curves and we care about lines. So with the same optimizer, without retraining, we can actually get all of these odd shapes and features from your detections. So what are the challenges that we have right now? As you can see, the crosses are not always working, and this is because of the way we build the edges. As I said, we build the simplest one that doesn't allow any crossing. So in this case, maybe that would be a solution. We also might have a problem that the detections might be so sparse you actually don't incorporate all of the edges, all of the object. And the truth is that we would like to hear from you. We want to hear what challenges do you have and what are the challenges of your data set that we could potentially help with our framework. So what's next? We made the repo public. So if you want to have a look, have a browse around, this is the GitHub for it. And the idea is that we made it very versatile and that we can play around. So the first point is that we can actually we can change the images and the detections that you have, because as I said, we just want the data that already exists. So we can consider tomograms, we can consider time series, anything that we can then build into a graph. We can have different types of networks for the extraction. In this case, it was a ResNet. Maybe that's not the best one to retrieve the images that we care about. We can also have other types of graph networks, and we can have fancier graph neural networks that learn about the local context, but as well keeping the attention on. And we can try other optimization algorithms. But what I leave you with is that we are fine-tuning the pipeline. We're trying to make it all work from the detections and your data all the way to the object separation. We want to have data augmentation for all of the stages to improve the results. We want to experiment, and we really just want to address the GitHub issues that we currently do have. So yeah, that is all from me. Thank you very much for listening, and I will take any questions. And I want to thank everyone that has been helping us, not just with this project, but all of the cryoEM work at the Turing. So thank you.